Good day everyone, welcome to the course on biological data analysis and visualization with R. In the last few classes we have talked about basic statistical tests starting with t-test etc., and then we followed that up with correlation analysis. We have talked about Pearson's correlation and Spearman's correlation, and we have seen how to do basic XY plots, and then we can also customize these plots. In this class, we will be talking about another statistical method that is very widely used in biological data analysis. Is called analysis of variance or ANOVA. Okay, so these are the topics that we will be talking about here. So, first is analysis of variance ANOVA, and then we will talk about multivariate analysis of variance or MANOVA. Okay. So, we will talk about ANOVA in much more detail, and multivariate analysis is simply an extension of that, where we have multiple dependent variables. Okay, so analysis of variance, what it does, it kind of takes the t-test and it kind of expand these to more than two groups. Okay, so we have talked about t-test where we are comparing between two groups, and ANOVA allows us to do this comparison across many groups, okay, more than two groups. Okay. And like t-test, what we are doing here, we are looking for difference of means between these groups. In t-test, we are looking at difference between means of two groups. Here will be difference of means between more than two groups. And what we calculate is something called total variance, which is contribution from different factors. So, without going into the mathematics of ANOVA, we will just briefly understand how what we are dealing with, right, and what kind of insights it can generate for us. So, in ANOVA we have one dependent variable, right. So, that is the something we are predicting or something we are interested in, and one or more independent variables, right, that actually determine the outcome or the dependent variable. So, we can classify ANOVA into different classes. We can do it in different ways. We can say we can have one way ANOVA or two or multi-way ANOVA, right. So, again there might be different names. So, most commons are the one way or two way ANOVAs. So, one way ANOVA we have one independent variable, whereas two way ANOVA we have two independent variables, and you can have more than two independent variables as well. Okay, so, again and so we can have one way ANOVA or two way ANOVA, but we also have we can classify ANOVA in different ways. So, one is fixed effects model, the second is random effects model, and the third one is mixed effects model. So, I will explain now what are these different types of models, what are fixed effects model, random effects model, and mixed effect models. So, fixed effect model is where the independent variable can take only a specific set of values. Okay. So, here is an example which helps us understand. So, in this example, right, this is a table, again we are looking at this drug dose versus efficacy, right, and the efficacy is the dependent variable, right. This is the outcome that we are interested in, and we want to measure, and we want to see how this changes when we change the drug dose. Okay. So, the drug dose here we are doing this treatment, and we said that at specific levels, right, we have this 5, 10, 15, 20. So, this is fixed effects model, right. The independent variable drug dose here can take only a specific set of values. Okay. It is not like a random values, it cannot take random values, because we have set these values. So, we do the experiment, we set these values. On the other hand, random effects model, the independent variable can take a range of values, okay, and these are not fixed. So, for example, we are testing this effect of drug dose, right, or efficacy of this drug dose of this drug in a population, right, of individuals, and of course, we are not fixing. For example, we want to estimate what is the effect of age, right, how does it affect the efficacy of the drug. Okay. So, now age is an independent variable that can take a range of values, right. This is something it is not fixed. Okay. So, it is not like we are fixing the drug dose, the treatment, right. So, this is a random effects model, okay, where it can take a range of values. So, this is something that we will encounter in biological data, right. So, in many cases if we have, let us say, field data or clinical data, we want to estimate the effect of patient age or maybe patient blood sugar, etc. Those will have a range of values. Whereas, in some cases we are trying to test effect of a drug, right, maybe we will have a specific values of the drug dose, right. So, we will have these kind of situations in real world when we deal with biological data sets. So, we have talked about fixed effects model and random effects model. So, we also have mixed effect models, right. So, where we have fixed effects variable plus random effects variable. So, where for example, in this case we have one independent variable that can take in a range of values, right, which is the age here, it can take a range of values, whereas the other independent variable, the drug dose, it can take only specific values, because these are said by us, right, those who are doing the experiment. Okay. So, mixed effect models, you will have this kind of variables, different types of variables, right, that can take fixed values and that can take random values. Okay. So, here we have the efficacy, right, that is our dependent variable, and we have these two independent variables. The goal of ANOVA is to test, right, whether how the drug dose and the age actually affects efficacy. Okay. So, we can have these two ANOVA, right, we have two independent variables, and we can test for effect of drug dose as well as the effect of age. We can also look for interaction between drug dose and age, right. So, what does that interaction tell us is whether there is specific drug dose that works best for certain age range, etc. That kind of analysis can also be done. Okay. So, we have talked about the basics of ANOVA. Again, when you do ANOVA, it will tell you whether any factor is actually important, right, whether it is one way ANOVA, two way ANOVA, whether any factor has important effect on the dependent variables, right. Again, we will get an effect size and the p values, level of significance, and based on which we can kind of assess the effect, right, of a specific independent variable. So, MANOVA is an extension of ANOVA, right. So, here in ANOVA we are trying to predict effect of independent variables on one single dependent variable, right. So, this efficacy is the single dependent variable in this data. We can have multiple dependent variables, and this is when we will do the MANOVA, right, which is the multivariate analysis of variance. Okay. So, in this example we now add another dependent variable, which is, let us say, blood pressure after treatment. Let us say we are testing a drug dose for a drug that actually affects blood pressure. So, we can have this blood pressure after treatment, and these are the specific values that we can take, right. So, this is again we want to, let us say, see how the blood pressure is affected by drug dose, or how the age of the patient or age of the person actually affects this blood pressure, right. So, we can have multiple dependent variables. You can imagine in your case, right, there are specific data sets where we can we have to predict, right, multiple dependent variables. Okay. So, we'll talk about ANOVA mostly rather than MANOVA, and we'll see, right, some of these functions that are there. So, if you want to do this analysis ANOVA, we can use the LM function. This actually builds a linear model. We will use this a bit later when we talk about linear regression. We will use the LM function. So, this builds a linear model. The specific function in R to do ANOVA is the AOV function. Okay. So, this is actually built for ANOVA, and we will see how this works. We can do MANOVA in R as well, it is the MANOVA function. Okay. And then we can look into summary of any model, right, ANOVA model that we have built using the summary.AOV function. Okay. So, with that discussion, we can now go into the hands on, right, and we can try out this ANOVA analysis on our data set. Again, we will stick to the same data set, because we have now introduced this data set, and we are familiar with the data set, and we can run a lot more analysis. So, here is the data again, right. So, we are using this data, which is the filtered data in IST, header equals to true. We have already described the data. So, this data contains gene expression measurements in two different environments, as well as gene expression variation among cells within a population, right, in again in two different environments. Okay. So, we have average expression data as well as heterogeneity data. Okay. So, just to remind you, these are the names of the columns, and in the last class we have seen this, right. The first column is gene name, then we have the average expression, then we have the DM values. These are the heterogeneity measurements. We said like this is single cell measurement, and high DM would mean high variation among individual cells, and low DM would mean low values, right. So, these DM values can take positive values as well as negative values. Negative values would mean low variation. Then following that, right, after this sixth column, you have lot of features that are trying to predict what is the average expression and the heterogeneity measure, right. So, that is the idea of this data. Can we predict from these different features the average expression of a gene as well as its heterogeneity among a cell population. Okay. So, these features include, for example, the nucleosome occupancy, transcription factor binding sites, how many transcription factors are bound, so on and so forth. And here probably, for example, you have the histone modification patterns, you have a lot of for example, the modification on the DNA, like methylation, acetylation, etc. So, all those things are considered, and this is a large list of features that have been compiled from the literature. So, the goal of this is to actually now see what are the factors actually affect, for example, the average expression or the heterogeneity. Okay. So, we will pick only certain features rather than all of this. Of course, doing this on all does not make sense, right. ANOVA is not the best way to do this, right. So, we will see maybe two features and see how they are able to predict the gene expression of the heterogeneity. Okay. So, let us see for ANOVA model, right. So, we have this, let us say, model, and we will use this function AOV, right, and let us say we want to predict first the average expression. Okay. And so, when we write this data $average expression YPD, this is our dependent variable, right, and we want to now check effect of two independent variables on this dependent variable, right, whether whether these two independent variables affect average expression. What are those independent variables? Data $. Let us take any of these two, maybe this one, data box, whether data box is present or not, and let us say one of the features we know there could be some effect, right, is the number of regulatory TFs. Okay. So, this is the ANOVA model we have defined, and after we run the model, we will assign this to this model value, right. So, we have defined a variable where we will store these results of this ANOVA analysis. Okay. So, here we have AOV, right, that is the command, dedicated command. We can also use LM, and then we have the dependent variable first, this tilde sign, that is telling, right, this test for like how these independent variables affect this dependent variable, right. And here we have taken two independent variables just as an example. You can try out any variable that you like from this data, and we run this. Okay. So, this has run now. Let us see model. So, it is saying, okay, what are the sum of squares that we have, degrees of freedom, etc., residuals. So, here we are not getting anything. So, what we will get is if we run summary dot AOV model. Okay. So, I will run this here again, right. So, what you see here is the degrees of freedom, sum of squares, mean square, f value. Here it is better now, we can see this in one column. So, it is actually estimating the effect of each of these factors. Okay. So, these are the independent variables, right. So, data box, whether data box is present or not, right. So, data box will have only yes, no, 0 or 1, you will see that. So, it does not have any exact value. So, we will see that in a moment. Then we also have this number of regulatory TF. This is a variable that can take any value, right. So, data box is fixed effect, right, it is either 0 or 1. it cannot take 0.5, it cannot take 2, right, it is just yes no. Whereas number of regulatory TFs, it can take any value. Okay. So this number can vary from 0 to 10, 15, etc. right. Now what you see here is this is the f value and this is the probability, right, this is the p value that we get, that these factors, these features, these independent variables have some effect on this data, on this dependent variable, which is the average expression that we defined. Okay. And these effects are significant now, right. So these effects are actually significant on this data set, right. So there is significant effect on the data set. Okay. So this is something we can do. We can also now see, right, whether these parameters have effect on the heterogeneity, the DM measure, right. And okay, so we can run, let us say, just to distinguish between the two models as model 2. Okay. And again we have summary dot AOV model 2 and here again we have the values, right, and you see the p values here, they are extremely significant, right. So this is something that means these features are actually affecting this heterogeneity measure as well. These are also important features, right. So when you look into this data box, right, so this, because this can take only 0 or 1 value, right, so which means there is significant difference between the groups, right. So whether you have data box or not, that matters a lot for heterogeneity as well as average expression. And similarly for this one, number of regulatory TFs, it's kind of saying, okay, as number of regulatory TFs change, it actually also change the mean expression as well as the heterogeneity measure. Okay, so this is simple two-way ANOVA. So we can also try, for example, interaction between terms, right, between these features. Okay. So what do I mean by interaction is, in some cases you will have this data box and then you will transcription factor, the number of transcription factor will vary, right. So what we are looking at here is the data box times the whether there is any interaction between these terms, right. So when you have data box and the number of regulatory TFs is high or low, whether that matters or not. Okay, so this is the interaction term, and so to test for interaction terms, what we do is, for example, we say data box multiplied with data number of regulatory TFs, right. So we have these two independent variables that we use with this multiplication sign. So which means the ANOVA model now knows that you also want to test for this interaction between these two independent variables. Okay. So let us distinguish this as model 3 here. Okay. And we can again check this summary.AOV model 3. Okay. So now we have 3 rows here, you can see these 3 results. First 2 are for these individual factors and the third one is for interaction between these 2 factors. Okay. So it means this interaction is also important, right. So whether you have when you have data box and when you have the number of regulatory TFs varying, right, that combination is also important. Okay. So we also sometimes look for these combinations. Okay. So let us see if we maybe we want to explore this a little bit, right. So how do we explore this is, maybe we want to first look into this how data box and this DM YPD varies, right, and maybe we can use some plots here, very simple plots, and as before okay. So you can see this right here. Now the plot will be different because, as I said, data box you have only 0 and or 1 value, whereas the DMYPD value can change. Okay. Now this is not the best way to represent this data. we will see that when you have this kind of classification, these factors, how do you represent those data. So, the best way to represent this would be box plot, which we will discuss in one of the later classes, right, when we talk about more slightly more advanced visualization techniques. Okay, so we can check the other ones, for example, right, which we have already checked, of course, right. This is how the whole thing looks like. We can also look, for example, how the number of these regulatory TFs vary, right, and we can use this histogram, right, and we can see this how this number varies. So, in some for some genes that we do not have any transcription factor binding, either this is missing data, right, we do not have experimental data for this, or actually there is no binding, transcription factor binding. Okay. And for the rest, we see some transcription factor binding between, let us say, 10, 20 and etcetera, and there is also some genes for which we have very high number of transcription factor binding. Okay. So, we can see these values, and then we also do look at the ANOVA and we see this analysis. Okay. So, one of the things we probably also realize, right, this correlation also kind of captures this, right, there is some effect of number of regulatory TFs on the gene expression, average expression, as well as the heterogeneity pattern. Okay. So, this is for ANOVA. So, let us now look into MANOVA, right, if we can run MANOVA, and then we will see if we can run also LM, right. Although it is not preferable, we can start introduce the LM function also. So, let us see if we can MANOVA model, let us say. So, maybe we can say MANOVA model. So, okay, so what I will do is I will simply first write this MANOVA function, and okay. So, we can also try help MANOVA, right. When you try this help function, it will tell you how to use this, right, and here it will tell you, right, what are the arguments that you have to give, etcetera, right, and with some examples. Okay. So, here as you can see, this is in this example for here, let's zoom in a bit, right. Okay. So, what we are seeing is we have MANOVA, and we want to see, for example, this effect like predict two dependent variables here, right. So, in that case we have to give these two dependent variables like this using cbind. Okay. So, let us try this out, okay, and I'll go back to R now, and let us try this out. Is model new cbind, right. So, what we want to, what are the two dependent variables that we can use is the average expression and the heterogeneity, right. And then we can use the same set of independent variables that we have used. Okay. So, let us now see what are the results. Okay. So, model new is this. Let us try the summary. Okay. So, here is the summary of this MANOVA analysis. So, what MANOVA is doing is actually running these individual ANOVAs, right. So, first it will look for response variable 1, which we defined as dependent variable 1, right. So, that is the response variable 1 here, and these are the results for first response variable, which was the average expression, and then we have the second response variable, okay, and the results for the second response variable, which was the heterogeneity. So, what you see is like actually we get the same results, but we can run this analysis together using MANOVA rather than doing individual ANOVA analysis. Okay. So, so this is how we can run ANOVA models and MANOVA models, and we also have seen how we can interpret the results, right. So, we have also run the interaction analysis. We can similarly try this here, right, times. So, interaction between these two independent variables, and again we have the summary we can check, right, and again we see similar results, right. So, it is actually running ANOVA again, but for multiple dependent variables or response variables separately, okay, using the AOV function. Okay, so, last thing we want to try is just the LM function. You will see the output will be different, but most of the things will remain the same. You will get the same results, you will see the significant p values, etc. So, we will try this now, the model linear, okay, LM. So, LM will take the, we will try, let us say only one of them, the other one will remain the same, right. So, data dollar. So, we have the box plus okay. So, let us see what we have. So, this is LM model, linear model. Okay, so, if you write this model linear, it will just give us the model that is being fitted. We will talk about this in much more detail when you talk about linear regression. So, here is the summary of model linear. So, here the function is summary, not summary.aov. Its summary is saying this is the formula of the model that we have used. Okay, these are the residuals. These are the coefficients, okay, and these coefficients, the effects of each of these, right, that we have estimated here. And what you see is the results of the ANOVA actually here, okay, and below, which is the F statistic and the p value. Okay, so, p value is significant, right, highly significant. So, which means at least one of these factors affect the response variable, which is the DMYPD or the heterogeneity in expression. Okay. So, this is slightly indirect way of looking at this ANOVA analysis. So, we prefer to use the AOV function, and if you want to if you are doing the MANOVA, you will use the MANOVA function. Okay. So, we will use this LM function for linear regression. When you build linear models and do linear regression, we will look into this in much more detail. Okay, so, we can go back to the presentation now. So, we have demonstrated this hands on with these different functions, and we have seen how to run ANOVA and MANOVA in R. So, to conclude, we have talked about ANOVA here, analysis of variance. So, what it is doing is testing for effects of independent variables on one or more dependent variables, right. So, we have ANOVA or MANOVA. MANOVA, we have more dependent variables. We can do one way, two way ANOVA or even multi way ANOVA. We have also talked about fixed effects and random effects ANOVA, and we have actually done the hands on the real data set, and I have demonstrated, right, how we can run this analysis. So, I will encourage you to run this yourself, because I will share the data with you, and you can do this yourself, and you will be you will get a feel for these techniques through the hands on. Thank you.